Evolutionary Medicine in Zurich, Switzerland. So I welcome everybody to this seminar series of today. I will first shortly introduce the seminar series, and then I come to today's speaker. So in the Institute of Evolutionary Medicine in Zurich, every semester we have a seminar series about invited talks with local speakers, but also international speakers, where we discuss topics around medicine, but also evolutionary topics, so to bring together the two areas. This is our area of expertise. And so we come to today's speaker. This is Professor George Davey Smith, and he is the director of the MRC Integrative Epidemiology Unit from the University of Bristol in the UK. And I'm actually very proud and honored that he's with us today, as he's one of the most regarded epidemiologists today, and he was also former editor-in-chief of the International Journal of Epidemiology. And he will talk today to us about analogies between Mendelian randomization and natural selection. Mendelian randomization is one of his major research fields, and so I'm very much looking forward to his talk today. At the end of the talk, you will have the opportunity to ask questions to Professor Davey Smith by raising your virtual hand in Zoom. This talk will be recorded. And so now I will give over the word to George. And so please, George, you can share your screen and you have the word.
Great. So yeah, thanks very much for the invitation. I gave this title thinking I'd then find out something about the topic. And then, of course, I discovered I had a couple of weeks to give my talk. But I've enjoyed thinking about this. So I'm going to introduce a method used in epidemiology. I'm an epidemiologist, and therefore I'm interested in modifiable causes of disease that you can actually intervene on and do something about. And Mendelian randomization is an approach in epidemiology which attempts to establish the causal nature of associations that are observed in conventional observational epidemiological studies. So I'll introduce Mendelian randomization, and then I'll discuss some potential analogies with natural selection. And this is the outline of my talk, and you'll notice at the end, I've put for discussion, could MR methods in principle be useful in interrogating selection, and could they in practice? So I hope to learn from you in the discussion about the potential value of MR in this setting. So I'm going to start with a brief 2-minute animation, which introduces Mendelian randomization, and I'm going to go and get myself a cup of tea while it plays, as I've seen it quite a lot of times.
Epidemiologists are interested in understanding factors related to health and disease. Do smokers tend to die younger than non-smokers? It certainly looks that way. But disentangling cause and effect can be difficult. Smokers are different on average from non-smokers. They're more likely to drink heavily and have less healthy diets. But even if we measure these confounding factors, we may not measure them perfectly, or there may be others we haven't measured. Also, as people become ill, they may cut down or give up smoking, which could wrongly suggest that reduced smoking leads to worse health. We could randomly assign 50,000 people to smoke and 50,000 people to not smoke and follow them up to monitor their health. This removes the possibility that any other factor could be responsible for any differences we see in their long-term health. But this is neither ethical nor practical. Fortunately, we have all been recruited into an experiment without knowing it at the point at which we were conceived. Our genes, which are passed on randomly from generation to generation, influence how much we eat, sleep, drink, smoke, and more. These genetic influences are not affected by anything else you may or may not choose to do in your life. They're not related to confounding factors. We can use this knowledge to learn about cause and effect, grouping people according to their genetic code. This method is called Mendelian randomization. For example, smokers who carry one version of a gene called CRP tend to smoke less heavily than those who carry a different version. When we group people according to which version of this gene they have, we find that the people with the version of the gene associated with heavier smoking do indeed die younger. But is the gene influencing how long we live in some other way? We don't think so. When we look at the same gene in non-smokers, there's no effect on life expectancy, so the effect must be driven by smoking. Using this method, we can show that smoking causes lung cancer, heart and respiratory disease, and many other diseases, but does not seem to influence depression or anxiety. Mendelian randomization has already begun to tell us about factors that influence our risk of disease. Now we're using the same approach in other ways to look at several risk factors together and to look at what influences disease progression, which may help us to develop new treatments.
Well, so I should say new treatments. This schema for Mendelian randomization starts with thinking about a conventional observational study. In an observational study, you will measure the exposure, for example, circulating cholesterol levels and the outcome, coronary heart disease, and you'll relate the two. But there are, of course, common factors that could influence both the modifiable exposure and the outcome, confounding factors, such as behavioral factors, socioeconomic factors, environmental factors, other physiological factors, etc. And also the early stages of your disease process leading to coronary heart disease could influence what you think is an exposure. So the direction of causation could be from the developing disease process and your exposure rather than from your exposure to the outcome. It's often impossible to exclude confounding or reverse causation as an explanation for the observed association. If you have a genetic variable that relates to the modifiable exposure, and now there's very many common genetic variants that do indeed relate to circulating LDL cholesterol level, then that variant will not have the confounding or reverse causation that exists for conventional exposures. Obviously, your germline genetic variant can't be influenced by any confounding factor or by developing disease process. So that it can be referred to as an instrumental variable that influences your exposure but has no other effect on your outcome. And in that situation, you can take both your instrumental variable outcome association and the association between your instrumental variable and your modifiable exposure, and you can estimate the effect of the exposure on the outcome, which you consider to be a causal effect. The arrow becomes red in that situation. So that is the basic notion behind Mendelian randomization. It was, when postulated, it was put forward that it would be most powerful and would be truly analogous to a randomized trial in a between sibling design. So if you compare, if you're looking at genetic variants that differ between siblings and how they relate to the difference in outcome risk between siblings, there obviously can't be confounding by population stratification, ancestry, origin, etc., which might lead to distortions in just straightforward population genetic studies. But at the time, 20 years ago, there was no studies that were large enough at all to have those data, although now they do exist. And so Mendelian randomization now in its true form, its form that is directly analogous to a randomized controlled trial, is being carried out. Now this approach is being utilized to try and give evidence about a modifiable exposure that you might then want to test in a randomized trial, for example. If you're going to make that leap, then you have to consider that the exposure has the same effect on the outcome, whether it is modified by genetic factors, it's influenced by genetic factors, or it's influenced by environmental factors. Both will have the same effect on the outcome. So it doesn't matter whether your elevated cholesterol is due to the fact that you've been unlucky in the distribution of genotypes related to cholesterol, or that you eat a lot of sausages. Both will have the same effect on coronary heart disease in relation to how much they elevate the cholesterol level. So that principle, so the fundamental assumption there is of gene-environment equivalence, is that the downstream effects of a change in a phenotype are independent of what influences that phenotype. Now, this is reasonable, and I'll come back to this throughout the talk. This is a reasonable assumption. And if you consider sort of a condition called pellagra, which is due to niacin deficiency, we now know it's due to niacin deficiency. That wasn't known at the time. It was demonstrated to be due to a dietary deficiency. There's a monogenic Mendelian condition known as Hartnupp's disease, first noticed in the early 1950s, which is phenotypically very similar to pellagra. And Hartnupp's disease is caused by a genetic disorder in absorbing amino acids, including tryptophan, which is converted to niacin. And the niacin deficiency generated by the disorder leads to a phenotype very similar to the deprivation of niacin through dietary deficiency. And in developmental genetics, this is considered non-controversial in a way. Mary Jane West Eberhardt, in her 2003 masterpiece, refers to gene-environment equivalence and gene-environment interchangeability, the fact that gene expression is influenced by external factors. The downstream effects will be the same as if it is influenced by germline genetic differences. Zucker-Kendall and Willett in PNAS in 1988 wrote that no doubt all environmental effects can be mimicked by one or several mutations. I mean, thinking about that medically, I think for most sort of medical disorders, that makes sense. I mean, if you think of sort of outcomes like being hit by being run over by a lorry, then maybe not so much, although of course the genetic influences on risk-taking behavior will have the same ultimate effect as any differences in risk-taking behavior induced by other means. So this is an important assumption if you're going to make the assumption that you can use that evidence for thinking about modifiable factors. Now, in the sort of MR field, there's very many examples now of where you can demonstrate this by comparing Mendelian randomization studies with randomized controlled trials. So talking about cholesterol, the atherogenic portion, or is non-HDL cholesterol, as it's sometimes called, which mainly consists of LDL cholesterol and remnant cholesterol, that is lowered by very many drugs. Statins are the best known of such drugs, but there's also drugs which target other genes, like the Niemann-Pick C1-like-1 gene, which is involved in cholesterol absorption across the gut. And a drug called ezetimibe, which has been in randomized trials, also reduces coronary heart disease. It's PCSK9, which is another genetic gene which relates to cholesterol level. There's a drug which lowers cholesterol through targeting that gene, and others. And now in the Mendelian randomization situation, of course, people have experienced the effects of the gene right from the moment of conception. And certainly we know that from birth on, your genetic variation influences your circulating LDL cholesterol level. You can show that in cord blood samples. You can show it at age 4. You can then show it right the way through life. And so in the genetic situation, the exposure is lifetime. So the people who have higher cholesterol levels due to genetic variation will have had higher cholesterol level, on average throughout life. Whereas the drug trials lower cholesterol level for about five to 6 years. That's the average, that's the sort of duration of the drug trials. And of course, you wouldn't expect the same effect on reducing coronary heart disease risk from just 5 or 6 years lowering of cholesterol to lifetime lowering, because atherosclerosis is demonstrably a lifetime process. But this relative difference between the drug trials and the genetic studies is constant across whichever method you use for changing cholesterol level. The genetic predictions are larger than the what you see in the drug trial, but the drug trials get about 40% of it, recover about 40% of the effect. So that's just one example out of, there could be many, of where you can demonstrate the gene-bound equivalence through randomized trials compared to Mendelian randomization. Now, I referred to the genetic variants as being used in this setting as instrumental variables, the setup in epidemiology here is you're looking at X, whether X influences Y, but you have confounders, and then you have your genetic variant which relates to your exposure. And so this is the first of the 3 instrumental variable assumptions. This the 3 key assumptions is that your genetic variant isn't independent of X, or the relevance assumption, just that your genetic variant is reliably related to exposure. That's the one of the 3 assumptions, the only one that you can actually test directly. The others have to be defended on other grounds. The second instrumental variable assumption is of exchangeability or independence of confounders. And in this formulation, it is that the genetic variant, there's no common cause of both the outcome, coronary heart disease, and the genetic variant. Now, as the genetic variant is germline, there obviously at an individual level can't be a common cause, but there can be distributional phenomena, the most obvious of which is population stratification, whereby in some ancestral groups, the frequency of the genetic variant varies across ancestral groups, and those groups have different levels of disease for environmental reasons. And then the population of origin would be a conventional confounder. You can also generate confounding through what are known as dynastic effects. And these are that obviously in human populations, you don't just receive genetic variants from your parents, but you also receive the influence of the effect of those genetic variants in your parents, how those influence your environment. Now, for genetic variants in the parents that influence protein levels and cholesterol levels and most biological factors, basically, that will not have an influence on your environment. But if you're looking at genetic variants related to educational attainment, for example, as I've done in some Mendelian randomization studies, the variants will be received together with the fact that your parents will construct a different environment for you depending on upon the educational attainment genetic variants that they have. So this is why within family Mendelian randomization is additionally protected, because the confounding from population structure doesn't exist, and the confounding from parental environment won't exist if you do a between an analysis within sibships, so i.e. between the siblings. And the third assumption, which in instrumental variables terminology is known as the exclusion restriction, is that your genetic variant doesn't have an effect on your outcome except the effect mediated by the exposure of interest. And in genetic terms, this would be that there isn't any horizontal pleiotropy. So that obviously is a challenging assumption, and is the one which has sort of led to most work in Mendelian randomization, as you know, genetic variants can be pleiotropic. Now, in terms of the diagrams you see here are known as directed acyclic graphs, which are graphs where the arrows represent causal effects. The absence of arrows means that there is no causal effect between the two nodes or two letters here, which haven't got arrows between them. Directed acyclic graphs can be seen to have developed from path analysis, and that was, of course, introduced by Sewell Wright, also obviously a major contributor to evolutionary theory. And this is Wright's famous diagram from one of his early path analyses, where he was looking at the inheritance of coat patterning in guinea pigs, and the arrows here represent causal pathways. The difference diagrammatically is that path analyses allow you to have double-headed arrows when you don't know what the direction of cause is, for variables where it doesn't matter how they influence each other, because they are in a Markov process where you have variables that you have an ambiguous orientation of the arrows. And of course, in Sewell Wright's setup, it was genetic variation that provided the anchors, the causal anchors, for the forms of analysis he did. Indeed, Sewell Wright would have turned in his grave, I think, well, maybe before while he was still alive, he reacted to the way that path analyses were used in situations where you had no causal anchor, because the whole basis of them to him was that they were anchored to things where you knew unambiguously which way the arrow went. And these are just quotes from Wright early on, where he says, that the whole basis of the path coefficients, the fact that you can actually interpret them causally, comes from the evidence from Mendelian heredity. And he said this again and again, that this was what allows them to be interpreted causally. And virtually every analysis that he reported was anchored in this way. So to get to the sort of major issue, which is pleiotropy. Now, it's important to consider that some different potential modes of pleiotropy here. The one which it would distort your estimates from Mendelian randomization would be horizontal pleiotropy, by which one means that the pleiotropic path does not involve your exposure of interest. So this would be a genetic variant that influences both cholesterol level and some other factor through diverging pathways. So its effects on cholesterol level would be independent of its effect on any other potential source of variation in your outcome. Note that to actually distort your estimate, it isn't just that the genetic variant needs to be horizontally pleiotropic, i.e. influence other phenotypes. It has to be that phenotype in turn influences your outcome. Your genetic variant influencing cholesterol level could influence any other factor that doesn't itself influence coronary heart disease without that distorting your estimate. Your estimate would only be distorted if the genetic variant influenced a factor which is itself a causal factor for coronary heart disease, i.e. there was a complete pathway between the genetic variant and coronary heart disease, which isn't through cholesterol level. Now, vertical pleiotropy won't be a problem if it's this form of linear sort of pleiotropy where your genetic variant influences another a phenotype, which then influences cholesterol level, as long as that phenotype itself doesn't have an influence on coronary heart disease. So vertical pleiotropy, which is obviously constitutes a sort of major proportion of where a single genotype influences many phenomena, that will often be through this vertical situation. Now, so you can think of any genetic variant which influences body mass index will then influence a huge number of outcomes, because so many outcomes are influenced by BMI. Like 50% of metabolites are influenced by body mass index, and all these many other things will be influenced by BMI. But in terms of estimating the causal effects of body mass index, those are, of course, downstream causal effects of BMI. So they would come into play however you influenced your BMI was influenced. So vertical pleiotropy in itself isn't a problem for Mendelian randomization. It is a problem, though, if you don't know what the primary phenotype is. If rather than considering that your genetic variant for body mass index, if you consider that it is a genetic variant for some factors which are downstream of body mass index, and then you interpret the causal effect as being due to that downstream factor, then you will get the wrong answer, because of course everything that body mass index influences will be associated with your outcome, but not because it causes your outcome, but because body mass index is a confounder. So that is a problem. So if you imagine here that X, let's consider X to be a protein, which I'll discuss later, called C-reactive protein, which is an acute phase reactant. Everything bad puts it up, including atherosclerosis that is developing. But body mass index influences CRP, and one of the strongest genetic associations with CRP is the FTO variant, which influences body mass index because of this downstream effect. If you mistakenly think that the primary phenotype associated with FTO is C-reactive protein, you then get the wrong answer. So if you don't know what the primary phenotype is, the one which comes first in the chain of factors that might influence your outcome, you get the wrong answer. And this is something which distorts interpretations of quite a few published Mendelian randomization studies. And then more problematic is when you have correlated pleiotropy, which is that this primary phenotype influences both your exposure and your outcome. So that situation is particularly difficult to deal with. Now, there's many threats to validity of Mendelian randomization, and there's long reviews written about them. I mentioned population stratification. There's also the fact that because the genes are assigned at conception, if there's an influence of the genetic variant during the fetal development, for example, which leads to adaptation during development to a different sort of level of exposure, then that will mean that manipulating your exposure later down the line will produce different effects to the ones seen in the genetic association. But I'll focus on pleiotropy because this is probably the major factor that leads to incorrect interpretations. So the horizontal pleiotropy could be balanced in the bias and the causal estimate from the various SNPs balances out with a mean of 0, or it might not be balanced. If it's not balanced, then it'll distort your estimate. Obviously, when you have multiple genetic variants, this can help with your interpretation because it's unlikely that all the genetic variants balanced out in the same way. So here's a whole stack of SNPs related to cholesterol level, which relate to coronary heart disease to the extent it's anticipated by their relationship to cholesterol level. In a situation where this happens, you'll also not see any heterogeneity in your causal effect estimates, and so the heterogeneity statistic is key. There's a whole host of sensitivity analyses, which I won't go into, which allow you to relax these assumptions. There's been a huge industry in producing these sensitivity analyses over about the last 10 years, but I won't go into those. Instead, I'll now discuss the analogies between Mendelian randomization, setup, and selection. You can think of Mendelian randomization as reverse genetics, i.e. genotype first. So reverse genetics is when you sort of go from genotype to phenotype. You know what the genotype does, and then you look at the chains of phenotypes and understand how phenotypes relate or indeed influence downstream phenotypes. Selection, in a way, can be considered forward genetics because selection is phenotype first. I mean, as Haldane and people well before Haldane reiterated again and again and again, selection has to be through phenotype, especially in times when you couldn't actually assess a genotype. It's the phenotype that's produced that actually leads to the selection. In a case-controlled genetic association study, saying the massive studies that exist of coronary heart disease, you look at the cases of people who've got coronary heart disease, and because cholesterol influences coronary heart disease, you are selecting people with coronary heart disease. You select a group of people, a group for whom there is enrichment with variants which are related to higher LDL cholesterol. So that's what you get, and that's why when you do a genome-wide association study of coronary heart disease, lots and lots of genes related to cholesterol levels come out. Again, in evolution, selection is clearly by phenotype, but the phenotype drags the associated genotype to be at an increased prevalence in future generations. So it's the phenotype which drags along the genotypes. And the gene environment equivalence is just built into many of the basic ways of thinking about this. If you take the Breeder's equation, which you all know much better than I do, you look at the covariance of relative fitness, which will be some sort of latent construct, with your trait value, your trait of interest, and that's the selection differential. And the selection response is simply your narrow sense heritability multiplied by your selection differential. And of course, your heritability is your additive genetic variance divided by the total phenotypic variance. So if you're doing that calculation, you are assuming that the phenotypic variance, the component of the phenotype which is not influenced by genetics, the non-genetic component, has the same effect on selection as does the component which is influenced by genotype. And you can find quotes over 70 or 80 years which state this, and the same is the case for Robertson-Price equations, etc. Have the same assumption. So the gene environment equivalence assumption is shared by Mendelian randomization and by selection. With artificial selection, of course, the sort of mirroring of Mendelian randomization and selection is obvious. I mean, you select by a phenotype. So this is the famous Mays selection, Illinois Mays selection studies going on for 100 years, where both protein and oil have been selected for, high protein, high oil. And this is over 100 generations. You just take you breed from the plants, which have the highest protein content, and the protein goes up and up and up across generations. You switch after 55 generations, and you can breed up and then down, or down and then up. So you're selecting on high or low protein, and you can just switch the directions, and it's extraordinary data. And the same is seen for oil selection, but in the oil experiment, they did a switch back where they sort of went up, down, and then up again. So here, because you're selecting on oil, you drag along the genotypes which actually relate to higher oil level. So this is and this is of course cross-generation. So Mendelian randomization is within a generation. Usually there are some 2-generational Mendelian randomization studies. I don't know of any 3-generational ones, but selection is obviously a continuing process. I gave the title of this a long time ago, well, not to Nicole. At that time, there wasn't anything that I'd seen that pointed out at this analogy, but this appeared on the 25th of March for a paper in Scientific Reports, which just makes discusses how you can basically use Mendelian randomization to help improve rice yield potential. So so it's now the analogy has been reported elsewhere. So if we go to human studies, we can think of fitness as a phenotype in humans. Obviously, there's some, say, latent notion of reproductive value, the expected future contribution to the population, where the future can stretch forward a very long time. But of course, in studies that have actually been done, you have proxies for this, such as total number of live births, total number of offspring surviving to reproductive age, total number of grandchildren, age at menarche, earlier age at menarche, over historical time, being related to more kids, etc. So there's been this is just a handful of ones which have been used. But of course, you have the same issue as in Mendelian randomization, that factors which are downstream of the actual factor which is having the effect will be picked up in the same way as will that factor, and that you have to have other knowledge to say which is the one which matters. And Mendelian randomization can tell you which one, or help inform which one matters. So, for example, total number of grandchildren will mean that you get more birthday cards. If you have a vast number of grandchildren, you get a lot of birthday cards. But getting birthday cards isn't actually contributing to selection. Of course, polygenic estimates, again, you'll know this sort of literature better than I do, and also know the very considerable problems with some of this literature. You can look at factors which have genetic evidence of negative selection. This based upon the effect size in relation to allele frequency, and the factors which turn out. The top factor that comes out of this analysis is age at menopause, and I've emailed to check that direction is correct, because I would have predicted that the later age at menopause should give you a longer reproductive age, but later age at menarche would reduce that, and does indeed show evidence of shows evidence of negative selection. There's also external evidence based on educational attainment and number of children. So certainly for the last few generations, people who have higher educational attainment have lower numbers of kids. So this sort of throws up factors like that, and like age at first live birth. The older age at first live birth is also has signatures of negative selection. But obviously, pleiotropy is a very important issue in Mendelian randomization and in selection. So here's one of a couple of sort of early but quite influential Mendelian randomization findings. This related to C-reactive protein, which I mentioned, shows very reliable associations with coronary heart disease, very strong statistically, seen across huge numbers of studies. And 20 years ago, people were talking about C-reactive protein as the new cholesterol, and there was drug programs. Pharmaceutical development was aiming to develop things, pharmaceuticals that would lower CRP because it was thought that it possibly caused coronary heart disease. Interleukin 6 was also investigated, and you got very reliable associations in this triangle of these three factors, IL-6 being another circulating cytokine. Mendelian randomization with very specific instruments, i.e. cis variants in the CRP gene, or a gene in the a genetic variant in the IL-6 receptor, allowed this to be deconvoluted. So it's known that the causal direction is IL-6 influences both CRP, and IL-6 influences coronary heart disease. Early stages of coronary heart disease influence CRP, but here, of course, the fact that IL-6 influences CRP, if you interpreted CRP as being the causal factor, you would be wrong. And of course, similarly, other effects in selection, this would be. I might have dropped out my slide about effects on selection, but it might come later, so we'll see. If not, I'll say it at the end. But examples of that. Well, I mean, one always discussed is sort of antlers in stags. Is it because they are actually a sort of indicator of of other forms of beneficial development, which are actually the factor which is selective, rather than them actually being part of the process that leads to them being selected. Now, those are sort of simple situations. In many cases, you have much more complex paths which never get permanently deconvoluted, but using multivariable Mendelian randomization, where you can use genetic instruments for different traits, does allow them to be reliably sorted out in many situations. So, for example, HDL cholesterol, the strength of evidence on the apparent protective effect of HDL cholesterol in coronary heart disease was strong enough for it to get into common parlance as good cholesterol, and for literally billions of dollars to be spent on drug development and massive randomized trials of a whole host of ways of elevating HDL cholesterol, all of which produced no benefit. So despite some of the strongest epidemiological evidence there is in the cardiovascular field, what was being identified and was very strongly considered to be a causal factor was not in itself causal. That's circulating HDL cholesterol. And as I say, billions of dollars were invested in that. And Mendelian randomization, the first study appeared in 2006, before the first trial reported, consistently suggests there is no actual causal effect of HDL cholesterol on downstream phenomena. So I'm just going to finish with sort of what genetic variants related to phenotypes might sort of hint at, but also the problems of only having current data really on how genetic variants relate to phenotypes, only having the data, of course, that you can gather now, and making and trying to make any sort of historical inference is problematic. So probably one of the best known just so stories in evolution, still in all the textbooks, is that lactase persistence arose because it allowed people to drink milk, because people who are lactase non-persistent, so lactase is lost after weaning. That was definitely the ancestral genotype, and there's been very rapidly selected for genotype that allows lactase to persist, and you can carry on digesting lactose. And it was just, it's often stated that this actually allows milk consumption, but we have now a vast amount of data that demonstrates that doesn't. This is my interest in this, was that when the lactase persistent, the European variant was identified, we quickly genotyped it, and what was then a large population of about 4,000 elderly women across Britain, and was really surprised to find basically very weak associations with any milk phenotype, such as not never drinking milk or full fat milk drinker. This paper published in 2009. It was published that late because the reviewers said this can't be the case, because it was so well known that this had a major effect on milk consumption, produced major symptoms in people who tried to drink milk. But now it's been replicated in populations of well over 1 million, and that there is basically a small, very small in relation to the variance of milk use. And this is not a processed milk. This is not milk where the lactose has been partially removed. This is just ordinary milk with lactose. There's only there's small differences in milk consumption, really small differences between genotypes. So on the basis of those data, and we said that this did seem to put some doubt on the cultural historical hypothesis, saying that milk was generally nutritious, and therefore people who are lactase persistent could actually drink more of it, and therefore there was a nutritional advantage, and or other advantages such as calcium, vitamin D, etc. Didn't really seem to be the case, as people could drink milk anyway, and postulated instead that it was more likely that actually once milk was available, everyone drank it, but that those who are lactase non-persistent in an environment with heavy zoonotic disease burden would be selected against through disease like diarrheal disease, which of course was the commonest form of death of infants since we've had sort of statistics of this. And the ancient DNA data demonstrates that the increase of lactase persistence came thousands of years after the introduction of milk, and where there's evidence from pottery shards, etc., that milk was being widely used, and that one hypothesis here is that the selection only started occurring once communities were concentrated enough to have the circulation of infectious agents, which would lead to diarrheal disease, death, or other such diseases. So there was a very long gap between the rise of milk use and lactase and the rise of lactase persistence. But just one still sees that it said that selection at loci associated with a specific agricultural product ability to consume milk in Britain, for example. But the phenotype linked data now does suggest that it is not ability to consume milk, as does the fact that in China now there's the white revolution. In China has a very large proportion of the population who are all lactase non-persistent who drink milk. So one can sort of examine the phenotypic effects. Now you can get some evidence in that in obviously a much less severe disease environment, you do still see that lactase persistence is the strongest single genetic variant related to the gut microbiome differences, presumably through just hugely different levels of lactose in people who are lactase non-persistent. So you get some indication that this might have an effect on, or might have had an effect on diarrheal disease, etc. But you're sort of guessing, or just making some conclusions based on that. This is the PowerPoint that I was meant to have earlier when I talked about, we're talking about pleiotropy and MR and evolution. Now I know there's been doubt thrown on all aspects of the farm fox experiment, but famously when tame foxes were deliberately bred from over 40 years or more, the tails changed to more dog-like tails. Now that was unlikely to be what was selected for, because what was selected for was disposition, but was likely to be a downstream effect of that, but not the selected effect. Pointing to the need of trying to find ways of actually identifying what the actual selected effect, selected for phenotype was. And very recently, I then came across it. This is obviously a classic to you, and so you'll presumably think everything I've said is banal, because this had been this notion of actually using genetic anchoring to try and remove potential biases, confounding bias, etc. Although not with germline genetic variants, but by using the sort of Sewell-Wright's whole genotype type anchoring in breeding experiments was proposed by Shiner et al. In 2002 with a nice path diagram relating the W being fitness, E being the sort of environmental confounders, so looking rather like the Ivy graph. And interestingly, in that study, their estimates that used genetic causal anchors were larger than the ones that actually use the observational data, which is what you would anticipate, because of the fact that the genetic anchors relate to longer term differences, and also instrumental variables estimation corrects automatically for measurement error. So your expectation is of larger effect estimates in such studies. So it has been this has been utilized there. My acknowledgements. So to Jib Hamani, I discussed this with, I've discussed these issues with over quite a few years, and Sewell-Wright, who both introduced path analysis. He also was involved in the introduction of instrumental variables. His father, Philip Wright, is credited with that. Many people, he was an economist, many people thought that it was probably Sewell-Wright's idea, but rather pleasingly, work has now uncovered correspondence between Sewell-Wright and Philip Wright to show that they discussed it together and came up with it together. And of course, his Wright's work on evolution. So someone who contributed all the way. So that's it. Thank you.
Thank you very much, George, for this wonderful presentation. This was most interesting. And I'm sure we have many, many questions from the audience or remarks. So please, to help me out, raise your virtual hands if you would like to ask a question or have a remark or something. Jordan, go ahead.
Hey, thanks very much. This is a great presentation. So I'm a behavioral ecologist, and I was thinking your discussion of horizontal pleiotropy, and you really brought this up at the end, thinking about the breeder's equation. You know, it's an issue we face a lot when trying to differentiate direct selection on a trait like a behavioral trait and indirect selection just due to genetic or environmental correlations. And I was wondering if you could just comment on basically the relationship between the complexity of the trait and how useful you think randomization or Mendelian randomization methods are for detecting causality.
Yeah, that's a great question. Yeah, I think the simpler the trait, the better. Probably the area where Mendelian randomization is having the largest influence, and I think will continue to have the largest influence, is just studying the effects of proteins. I mean, that's of course, and there's sort of two complementary reasons for that. The first is that there's a simple trait, and you get simplish trait, and you can take cis variants, which are plausible instruments for the protein, sort of variants in the gene which encodes the protein, as in the case of the C-reactive protein. And secondly, they're druggable. You know, proteins are very good targets for modification. And then I think it's just straightforwardly, as you move from the sort of simplest, the simpler traits down, things become more problematic. So, I mean, I think Mendelian randomization has been very valuable in looking at traits like adiposity. But that, the complexity there are that we have measures like body mass index, which is which is a single measure of something which is clearly a complex phenomenon, because where the fat is carried influences the downstream effects. It isn't just a single measure of weight divided by height squared that does that. And those are sort of situations where you can get genetic variants that relate to different aspects of adiposity, and multivariable Mendelian randomization can be useful in teasing them apart. And then now some behavioral traits you have good instruments for, ones which I think have been very informative. They tend to be ones that where the name of the environmental factor is in the name of the gene, because which gives you quite a strong prior that there's something sensible going on. So alcohol dehydrogenase, which relates to differences at a group level, relates to differences in alcohol consumption, has been useful. Acetaldehyde dehydrogenase, which in East Asian populations, knockout, the null variant is common, leads to huge differences in alcohol consumption because of acetaldehyde production. Those studies have had really quite an influence on guidelines now about alcohol consumption. So the bad news for Mendelian randomization is that the apparently protective effect of alcohol on cardiovascular disease is spurious. If you use Mendelian randomization, suggests that it's basically little effect on coronary heart disease, certainly not any meaningful protective effect. It also shows that the effect that was the reason why alcohol was then said mechanistically to reduce the risk of coronary heart disease was because it raised HDL cholesterol, but Mendelian randomization shows that isn't good for you either, and it has a highly detrimental effect on stroke because of the blood pressure raising effects. So the answer there is the answers from the Mendelian randomization study are very different from the answers from conventional observational studies. I mean, the other behavioral trait is smoking, which can be reasonably well estimated if you're just looking at heaviness of smoking, because the nicotine receptor variant, which doesn't really influence whether you start smoking, but it has a strong influence on how much you smoke if you do smoke, and how difficult it is to give up. And that shows that smoking is even worse for you than the observational data suggest. But then if you're trying to look at very complex behavioral traits like risk-taking behavior, which, you know, there are attempts to do Mendelian randomization studies of that, but risk-taking behavior influences, you know, so many downstream pathways, from obvious risk-taking practices, to many of them, and trying to separate those is very challenging. So I think it's definitely can work for the more upstream, the sort of upstream and simpler traits.
Thanks a lot.
Thank you very much. Do we have more questions or remarks from the audience? Dave?
Really fantastic lecture. I by the way, have seen your earlier lecture that you posted on Twitter as well. Just really fantastic stuff. I'm not sure if you're familiar with my work. We're actually very interested in hypercholesterolemia as brought about by the ketogenic diet. And in particular, we're wanting to test to confirm that the individuals in our study do not have any genetic abnormalities that would have resulted in real familial hypercholesterolemia. Do you feel there's many who've been somewhat critical in that they feel Mendelian randomization has sufficiently shown causality of LDL cholesterol to the extent to where this study may not even be needed? Do you feel that there's enough data, there's enough strong data provided by Mendelian randomization studies on LDL cholesterol to feel confident that we should be seeing a likewise level of progression of atherosclerosis for the same likewise exposure that you would see with the genetic variants?
Yes, I mean, it's the one area where there's, you know, been sort of the largest number of sort of randomized individuals outside of vaccine trials. So there's been so many statin trials which are really homogeneous in their sort of predicted outcome. And then when you lower LDL cholesterol with PCSK9 inhibitors or Niemann-Pick C1 like 1 inhibitors, or even cholesterol ester transfer protein inhibitors, which only lower LDL cholesterol a bit, they raise HDL cholesterol enormously, and they were introduced for that. But they produce a small reduction in coronary heart disease, which is entirely, it's just a dot on the straight line that you would anticipate from their effect on ApoB. So LDL cholesterol, I think the Mendelian randomization studies really show that it's that ApoB, it's ApoB carrying lipoproteins, which includes LDL cholesterol is obviously the major contributor, but also some of the remnant cholesterols that have ApoB particles, possibly because the ApoB particle is necessary. The ApoB molecule is necessary for to get across the intima of the vessel, and allows that then to be cut to generate atherosclerosis. I mean, there's clearly other factors. I mean, coronary heart disease is multifactorial, but LDL cholesterol is definitely a very major contributor.
This population is at between heterozygous and homozygous FH levels, so it's literally like in the top 2-3% of compared to the general population. So presumably, as is the case with Brown and Goldstein going back to the 70s, if they were looking at homozygous FH children, I mean, the development seemed to be very, very rapid. That's why it's an understandable concern if it's diet induced, if this is a likelihood. So that's why we're interested.
Yeah, no, I think the sort of linearity of the the linearity of that effect is really observed in the trials as well. I mean, if you lower cholesterol in people who haven't got particularly high cholesterol, but they're at high risk for other reasons, you reduce their event rate by reducing their LDL cholesterol.
Thank you very much. Do we have more questions or remarks from the audience? So, yes, Adam, please.
Hey, yeah, I was just going to say it was really interesting, and I wasn't sure how you were going to draw this analogy between evolution and Mendelian randomization, but it was really, yeah, I thought it was good. I was actually going to ask the same question as Jordan originally, but so kind of going back to this lactose example, then it seems like the similar problem occurs with the horizontal pleiotropy to some extent, because the problem is that there's so much going on with milk drinking that we're not exactly sure what the effect was, even though it seems so simple that it's just, you know, an extra enzyme. But then actually because there's more to milk than just lactose, then yeah, it's actually hard to pull that to pull it apart and really know exactly what the selection pressure was. So I kind of understand, yeah, why going back to like the basic protein it seems to be the only like the really reliable thing there. So no, I was just going to say thank you for kind of awakening that concern and making me more cautious about these being able to draw these effects.
Thanks.
Thank you very much. Actually, I have another remark about this. George, you said, unfortunately, we have only the data from today. And what about ancient DNA analysis that we also do? Is this a help? Do you expect more information when we have more information about ancient populations? Because it's not so old, and this evolutionary pattern that is just a few thousand years, so we can have ancient DNA from this time. Do you expect more knowledge and better outcomes?
Yeah, no, I mean, I think the ancient DNA data is you know, completely fascinating, because it really does give you some notion of what you know, when selection was occurring. And you know, it was definitely a surprise to everyone. I think, well, most people, I think, when lactose persistence turned out to be rather more recent than I think most were postulating. And then you get this, you know, really, there's a really interesting story with fatty acid desaturases, where, you know, if you look at evidence for selection, you know, now at genetic evidence for selection, it's, you know, there's very clear evidence of this, and it's continuing. You know, it appears to be, but in different populations. Although I don't, I've only read reviews of this, so how robust the data are, I'm taking on trust. But you know, there's a suggested that it might be that the was selected for in actual opposite directions in Inuit and in other communities, but probably depending upon what was available dietetically. So in most places, it was the enzyme. So your inference has to be to the enzyme. All you know is that this enzyme is being selected for, as you know, Adam was just saying. And of course, the enzyme will have different effects depending what people are eating, basically. But the fact that you get these that it's selected for, but in opposite directions, is really interesting. The problem, the issue is that you can get ancient DNA, but you can't get ancient phenotyping. So you've sort of got to assume that you've got to then make assumptions about how will those phenotypes be be the same, similar, or are they likely to be similar? Now, I think with the sort of FADS variant and all the experimental studies on how this influences metabolism of different forms of fat intake, you'd think that's rather unlikely to have changed. But you have to make assumptions about about that, and then clearly things like what's been demonstrated with educational attainment, genetic scores related to educational attainment and intelligence, etc. They obviously could have had very different influences at different historical times, for which I think it's very difficult to make any to make very sensible conclusions about. But no, the ancient DNA data, as it builds up, is just really fascinating.
Thank you very much. Is somebody else who has a question or remark? I don't see. So I would like to thank you again very, very much for joining us, George. This was very interesting. And I like the audience for joining us all. And I wish you all a nice evening. So thank you very much again for this. And see us all hopefully in 2 weeks for the next seminar in evolutionary medicine. Thank